Hold it. You telling me there's a free webinar where I can learn about protein design and Foldit? Yes, I am. So I'm super excited to finally get to spill the beans about the IUBMB Trainee Initiative's first event.
So it's been super duper hard to keep this a secret as we've been planning it because I'm just so excited about it. So we're going to be having a webinar about protein structure prediction and design, both in like the theory, the practical, and how you can get involved with it in this game called Foldit, which, although it's a computer game, it's also scientific, and you're playing with it is actually helping scientists learn how proteins fold and function and how we can design new proteins to do cool things. So Drs. Baker and Koepnick are both at the Institute for Protein Design, the University of Washington, in the Baker Lab. And so David Baker is really one of the pioneers of protein structural prediction and design. His lab is responsible for Rosetta, Rosetta Fold. He's now applying these tools to do cool things. So he's not just looking at trying to model individual protein structures, but also the structures of protein-protein complexes, which, as someone who studies structural biology, knows that that is like a really big deal. And he's also looking at how like you can introduce modeling as long with the like crystallographic and low quality cryo-EM data to try to do things like that. And so I think that's really, really interesting. So he's going to tell us more though about his work on designing proteins from scratch and what's at the cutting edge of what's coming, and so how he's working to use these principles that he's discovered throughout his work with trying to model protein structures to actually come up with protein sequences that are going to fold into specific shapes that can do specific things, maybe bind drugs, bind to other proteins. So much cool stuff going on, and he's going to tell us about it. Brian Koepnick is going to tell us about Foldit. And so Foldit is this game that anybody can play. It's totally free, it's totally open. And basically what you do is you design proteins and you do like protein modeling stuff, but it's totally like gamified. And so the things that you're doing are actually going back though to the scientists to help them learn how to update their structural prediction models, how to design proteins from scratch. They did this, they had this paper that's really cool that they actually show that when you have people that, so if you're trying to predict a protein structure or like model a protein or whatever, there's just so many possible combinations, and computer algorithms can do a good job sometimes, but sometimes they only try like the conventional stuff and then they get stuck in like a rut. But they found that the creativity of people really helped them find new solutions, design like totally novel proteins. And then they actually they actually solved crystal structures of some of these proteins, and they look like exactly like what they were modeled. So some really, really cool stuff. And they have like a totally updated user interface and things that make it really, really usable to use. That sounded weird, but that really, it all integrates the actual principles though behind the folding. So the hydrogen bonding, the hydrophobic residues, the angle strain, all of these various things come into play, but in a way that you don't have to have a science background. And so they found that like most people who play the game don't have a science background. And so anybody can get involved, but there are also a lot of scientists involved, but scientists at all level. So here's what's on tap. So we're super duper grateful that Dr. David Baker is going to wow us for 30 minutes with the talk titled The Coming of Age of De Novo Protein Design, where he's going to tell us all about his work in this area and the basic principles that go into designing novel proteins, as well as some of the real life applications that have come to play with therapeutics and that sort of thing, as well as what's coming next, what's in the pipeline. And then he's going to let us ask him some questions. So come with those questions. And I'm sure there'll be many more after his talk, many more from, I will have to keep, I will have to let be quiet so that other people can ask questions because I've just got so many. But we're really grateful that he's giving us this time. So that'll be about 30 minutes of talk, 10 or so of questions, and then it's on to Brian. And so Dr. Brian Koepnick is going to be telling us about Foldit. And so he's going to tell us about how Foldit was used, how to help allow people to help contribute to fighting the pandemic by designing binders of SARS-CoV-2, like proteins. So they don't just do protein modeling, they also model like make small molecule modeling to design small molecules that bind to proteins, then bind proteins that bind to proteins, and proteins that bind to small molecules. So there's all sorts of things in Foldit going on. And so I'm really, really excited because he's going to tell us about this COVID-19 project. He's also going to demo it for us and show us how we can get started with Foldit, and how the interface works, show us an example, walk us through that sort of thing, so that we don't have to come in here knowing how to use it. And so this is, if you're familiar with Foldit, that's great. There'll be cool information for you too, if you're totally unfamiliar with Foldit, or if you're curious about Foldit, but you've never tried it. And so that's kind of where I am. So he's gonna tell us about the COVID-19 project, as well as like demo things out for us, and then let us ask lots and lots of questions. And so we're just so grateful that these amazing scientists are giving us some of their time, and we really, really hope that you'll join us there. So this is going to be March 30th, 9:30 Pacific Standard Time. It's going to be virtual via Zoom. We hope that you'll be able to stay the whole time, but if not, we totally understand. But that's the basic layout of the schedule. We don't have a registration date set up yet, but it'll be coming. So this is kind of like your teaser post. So follow the IUBMB Trainee initiative in order to stay up to date and to have the latest as soon as we have everything all set up. We'll allow you to register for this event. It's totally free, but we are asking people to register ahead of time, just to keep things civil and stuff. So everybody, this is your save the date, and we really hope that we'll see you there. So the IUBMB Trainee Initiative is behind all of this, and we're really excited about this. This event is spearheaded by the FEBS group. So if anybody's interested in learning more about the IUBMB Trainee Initiative, I have done posts on them. We are a group of trainees. So we are students and postdocs who want to help other students and postdocs to really get engaged in the biochemistry and molecular biology communities, share access to resources, fun events like this, and really just build a sense of community and help one another, because we're all in this together, and we want this shared love of biochemistry and molecular biology. And so we hope that you'll join us, and you don't have to be a trainee to come to this event, but we really do encourage trainees to come and ask questions. But we encourage everyone to come and ask questions. So hope we'll see you there. And I know I'll be there. I'm super duper excited and grateful for this opportunity. So thanks again to Drs. Baker and Koepnick. And I hope that we will see the rest of you there. And so stay tuned for that registration info. And thank you all.